Hello, everyone. I want to thank the organizers for giving me the opportunity to present some of the work that is being developed within the lab I belong to. I'm Alejandro Salinas-Medina. I'm a PhD candidate from the School of Computer Science in McGill University, and I will be presenting this project now. So I'd like to start by introducing some preliminary concepts that we are trying to mainly reach with the development of this project. That is some technique limitations within classic segmentation algorithms. So we're trying to map a segmentation framework with a simulation framework. We are trying to map these limitations, and we're also introducing foundational models within the segmentation in order to be able to leverage this model as much as possible. And also, we are building the multi-scale graph-based neural field equation framework that we are enriching with morphologic mesh-aware. We'll talk about that in a moment. So as a computer scientist, I'm trying to build a framework that it's domain agnostic. So for the neuroscience use case, I am using multiple modalities of the big brain, where, as we know it, there are some challenges that need to be tackled, like computer complexity, system-level complexity, and cross-model integration. So existing segmentation and simulation methods are not designed to be multi-scale or multimodal data and cannot be integrated together. So we have some challenges like the multimodal data world that we now have nowadays, where we have the chance to leverage a lot of different kinds of multimodal data. We have computational bottlenecks for dealing with big data. Disconnected pipelines, we're trying to build a framework that bridges the micro, meso, and macro scale, and also with the techniques as segmentation and simulation environments. So we're trying to tackle this challenge, and as I said, we are dealing with multi-scalability. So some of the framework goals is to bridge, as I say, the micrometer scale segmentation, high-performance computing, biologically grounded simulation. This is in order to have a unified, scalable framework that we're calling it structure-driven modeling. And all this framework has been called ScaleNet. It established a unified simulation segmentation workflow. So let's see the overview of this framework. We have different spatial scales that we tackle across different architectural layers. So the first one we'll talk about is the micrometer resolution structure. Here we introduce the first submodel of the framework that is called sex scale. We presented this framework at the IEEE Engineering in Medicine and Biology Conference at Copenhagen this year as a full contributed paper. So what it mainly does in summary is we have our 3D data set. We slice our 3D data set. Then within each slice of the 3D within each slice of the 3D data set, we take different tiles using a diffusion model that it's called the peronomaltic model that follows the same differential equation as the heat equation. We we diffuse the data in order to only leave one minimal region per neuron or cell. And then we apply a segmentation. So there are we have been able to segment a whole slice of the big brain with this quality within the segmentations. And we are using two kinds of segmentation. As I say, we're using a foundational model as the segment anything model. Well, in this image, you can see that the SAM segmentation is being performed under a zero-shot setting. This means that no prior knowledge is being passed to the model, and it's having these results that, as you can see, they are not good results. It's missing some cells or neurons, and it's maybe over-segmenting some regions. So we compare the foundational model segmentation with a customized region growing segmentation that works similar to a watershed segmentation, that it's a pretty basic segmentation technique. And the best results we got were two, where we used the foundational model with prompted. What I mean by prompted, that we use the identified centroids of the cells as prompts to the neuron, to the model, and then it produced better results. And well, the customized region growing, these both algorithms had similar results, as you can see in the equations, in the table and graph, with our customized region growing pipeline surpassing by a little bit the foundational model, the prompted foundational model. Okay. So next we'll talk about the next spatial scale that the whole ScaleNet framework tackles. That this is the region-specific simulation. So we are using a generalized framework for simulating distributed dynamical systems, where we have 2 dimensions, time and temporal and spatial dimensions. We represent with a mesh that it's a BigBrain mesh, surface mesh. And we have the evolution equation that this actually comes from the virtual brain. This is the equation within that framework, the virtual brain, that we use it within this framework for the dynamical simulation. We use it as a dynamical simulation engine. And finally, let's check the cross-domain integration layer, where we will be unifying our 2 previous spatial scales. Well, for this, we're trying to tackle the lack of unified methodologies that seamlessly integrate the model-driven and data-driven paradigm. This is something I'm really interested in really be able to integrate the model and data-driven paradigm. So we have multiple challenges like scalability, usability, integration. So our solution is, as I've said, this multimodal integration through a graph-based modeling and morphologic-aware connectivity. So this is the framework flowchart. This framework is under review by reviewers for Nature Communication Biology. So as I said, we use different modalities of the BigBrain. We use 1 micrometer histology to segment the cells with the segmentation model that I previously described. Then we integrate those morphological, we extract some morphological features from the segmentation, and we group them by label, anatomical label. With this information, we embed them in a BigBrain high-resolution surface mesh. We are also using the eBrain's knowledge graph in order to extract, also by an anatomical label, the required connectivity within each case. So in this case, we are using the hippocampus, as it is one of the most described regions, and there's a lot of data available within my lab. And we integrate them in a multimodal data integration layer that then we input to the virtual brain. And as I said, we use this as a dynamic simulation engine, where we have simulated 3 kinds of neurodynamics, like EEG, MEG, and both. Then we develop a validation pipeline that mainly is divided into 2 submodels. We have a graphic and a visual validation, where in the visual layer, we expect to see some biomarkers from the simulated data, and these biomarkers are expected to be there in the hippocampus activity. This is for the EEG. And we do quantitative validation using empirical and the simulated data. So, well, the heart of the framework is the multimodal data integration layer, where we have a series of steps where first, well, we have a simple graph. This is the simple graph architecture. Then we have the intra-unit dynamics equation that we have described previously. Then we do this short-range interaction using geodesic distance and a minimum threshold. Then we perform the long-range coupling with morphological modulation. This is achieved also with a morphological aware mesh interpolation. We use the geometric mean in order to be able to embed our morphologic features within the vertices of each in the surface mesh. This is done by anatomical label. And finally, the validation framework that I have previously described. This, we can see the equation of the quantitative layer, where we are comparing correlation between empirical and simulated data in the domain, frequency domain. Well, we can see a matrix of the structural connectivity before and after enriching it with the morphologic aware weights. Also, we can see the same data, but in the surface mesh. Here is the key of this integration is the geometric mean, that we are integrating the area and eccentricity morphologic features extracted by the segmentation by using the anatomic label and using, well, as I said, the geometric mean. And let's see some results. These are, we can see these two images that are both simulate, well, one is the one from below, it's the empirical EEG, and the one from above, it's the simulated data. We see the, visually we can see a similar waveform. And we can see that we then calculate the correlation value across in the domain, in the frequency domain. We can see that when the box plot from the right, it is the unified framework validation. We can see that the means of the correlations are higher, but there is no free lunch. So as we get more correlation, more accuracy, we are introducing a little bit of noise to the simulation. We can see that by the form of the box plots. We can see the validation standalone framework results. Again, this is the quantitative validation, and the validation unified framework results that we can see a difference around 10% of more correlations between the empirical and the simulated data. So, to conclude, I would say that morphological enhancement improved the simulation accuracy. So the framework is able to integrate high-resolution segmentation with dynamic modeling. This allows us to aim for more biological realism frameworks. And there's also contributions for GPU-accelerated segmentation, that we will not be able to segment all the cells within a BigBrain slide without a GPU-based solution. And well, as I said, we are aiming to have this framework to be domain agnostic, so we want to also test it within material science and different fields of science and engineering. So for future work, as I said, we are aiming to have a scalable framework. So we are working on scale the current framework from the hippocampus to the hippocampus with the right hemisphere cortex mesh, all coming from the big brain. And we're also developing a neurodynamics agents pipeline where we are giving the tools to a LangChain agent to be able to run all of this with a prompt to automate different regions, different scales, to be able to automate all this process. Finally, I want to thank all my team that works with me really closely. I want to thank Alan, Steve, PJ, Annie, Jordan, and Andrea, and also HIBALL, the Secretaría de Ciencia, Humanidades, Tecnología y Innovación, that it's the Mexican Secretary of Science and Technology that funds my PhD. And you can contact me if you're interested. There's the QR for my personal webpage. And that's it. Thank you so much for your time.